Jane Morris, and in this presentation I'll talk about the approaches to GMO regulation that different countries have taken, looking also at some southern African countries. I can't cover everything on this topic, so I'll focus primarily on the background to the emergence of regulatory systems around the world, and I'll be paying particular attention to the definitions of a GMO and the triggers for regulation in different countries. This is particularly important because it has implications for regulation of some of the new breeding technologies that are currently taking off. If we look at the history of how molecular biology and genetic engineering developed, the basic tools were developed from the 1970s onwards. Already by 1975, leading researchers were concerned about the possibility of novel biohazards arising from the technology, and so the scientists themselves convened the Asilomar Conference, which led to non-binding guidelines on research and regulation of GMOs. These guidelines allowed research and development to continue and brought the science into the public eye. Subsequently, individual countries developed their own regulatory processes, and international bodies also became involved. As countries developed their legislation, two basic approaches emerged. Some countries focused on the characteristics of the GM product, so that regulation was triggered by the presence of novel traits in the product. Other countries based their regulation on the genetic modification process used to create the product, and some countries used a combination of the two approaches. Then, following the United Nations Convention on Biodiversity in 1992, efforts were made to develop a binding international protocol for the safe transfer, handling and use of GMOs. This led to the development of the Cartagena Protocol on Biosafety, which entered into force in 2003. The Cartagena Protocol regulates transboundary movement of GMOs, but it has also guided the development of national legislation in many countries. It defines a living modified organism, or LMO, rather than a genetically modified organism, or GMO, but in essence the two are the same. The definition of an LMO is strongly process-based rather than product-based. Currently, there are 170 parties to the protocol, including the European Union and most developing countries, but notably excluding the United States and Canada. So, here's the definition of an LMO in the Cartagena Protocol. An LMO means any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. The definition of modern biotechnology specifies in vitro nucleic acid techniques, including recombinant DNA and direct injection of nucleic acid into cells or organelles, and cell fusion beyond the taxonomic family that overcomes natural physiological barriers and are not techniques used in traditional breeding and selection. So here we can see that the criterion for an LMO applies irrespective of whether the resulting genotype or phenotype could have arisen naturally or not. It's primarily based on the techniques used. The criteria for an LMO also apply irrespective of the existence of novel traits in the resulting organism. The question is, does it really matter whether regulation is process or product-based? Well, different definitions in different countries can be problematic if the same organism is regarded as a GMO in one country but not in another. It leaves the developers of new crops in a very uncertain position if they don't know if their product will be regulated or not. This is now becoming a major issue with the development of new breeding technologies that are starting to blur the boundary between conventional organisms and GMOs. And unless some resolution is found, the issue is likely to impact on international trade regimes. But right now, many countries are struggling to decide how to resolve the issue. There are a whole range of new techniques that are under discussion, and I don't have time to explain all of them here. However, the one that has generated most interest is the use of site-directed nucleases, such as the CRISPR-Cas9 system, which allow for precision genome editing, and this system is already being widely adopted. Site-directed nucleases of this type can be used to make targeted changes in anything from one base pair to the introduction of much larger pieces of DNA. So, I'm going to give you a few examples of how some different countries have approached GMO regulation and how well placed their regulatory systems are to take on board some of the new techniques. We'll look at both developed and developing countries, countries that are party to the Cartagena Protocol and those that aren't, and we'll also look briefly at some Southern African countries. Let's start with Canada, which is not a party to the Cartagena Protocol. GMOs in Canada are governed under the Federal Regulatory Framework for Biotechnology, whereby responsibility is shared by Health Canada, the Canadian Food Inspection Agency, and Environment Canada. Canada is unique in having strictly product-based regulation, in that it regulates plants with novel traits rather than GMOs. The fact that the legislation is product-based rather than process-based means that GMOs and non-GMOs are subject to the same approval process. So, for instance, crops produced by mutation breeding are regarded as plants with novel traits in many instances. Canada has an enabling policy framework and has approved a wide range of applications. So the trigger for regulation is the novel trait, which means a characteristic that has been intentionally selected, created or introduced into a distinct, stable population of the same species through a specific genetic change and, based on valid scientific rationale, is not substantially equivalent in terms of its specific use and safety, both for the environment and for human health, to any characteristic of the same species in Canada. So based on this definition, we can see that Canada will have no trouble regulating the products of the new breeding technologies within the context of its current regulatory framework. Next, we look at the USA, which is also not a party to the protocol. The USA has a coordinated framework for the regulation of biotechnology in which the Environment Protection Agency, the US Department of Agriculture, and its Animal and Plant Health Inspection Service, as well as the Food and Drug Authority, all have a role to play. Officially, it's product-based regulation, triggered by there being a so-called intergeneric combination of genetic material in the product. But in fact, it also includes some process-based elements. The USA has no specific GMO legislation, so it's made use of elements of its existing legislation. So, for instance, the EPA regulates GMOs under rules for plant-incorporated protectants and assesses them as pesticides. APHIS, in turn, regulates them as plant pests and includes any GM plant with DNA from a listed plant pest. This includes Agrobacterium, which is commonly used in plant transformation, and the cauliflower mosaic virus, since this commonly provides the promoter sequence in transgenic plants. The FDA, on the other hand, doesn't have a specific trigger, but relies on a voluntary consultation process. However, it can remove from commerce any foods it deems unsafe. It's been acknowledged that the US system needs to be updated, and a strategy has been published which, amongst other issues, acknowledges the need to address the regulation of future products of biotechnology. So there's still work to be done to clarify the way forward. Let's look next at the European Union, which is a party to the Cartagena Protocol and was actively involved in the development of the protocol. Its GMO legislation is based on a series of directives originating from 1990. The legislation is largely process-based and refers to techniques of genetic modification, but excludes mutagenesis. Not surprisingly, given its active role in the development of the protocol, The definition is similar to the one used in the protocol. The European Commission makes decisions on approval of GMOs, taking into account advice from the European Food Safety Authority. However, for many years, no decisions were reached because of the need for the member states to reach agreement. To try to overcome this roadblock, in 2015, member states were given the right to opt out of cultivation of any GM crop authorised by the European Commission. This opt-out clause does not need to have any scientific basis, and up to now, at least 2/3 of EU countries have invoked the opt-out clause. Next slide. EU regulation has been strongly criticised as being not fit for purpose, considering the new technology developments, and it's been characterised all along by regulatory paralysis. Although there have been a number of studies from European scientific bodies recommending that some of the new breeding techniques should be excluded from legislation, the matter has been referred by France to the European Court of Justice, and in the meantime the situation is in legal limbo. In Europe, the influence of the NGOs is particularly strong, and decisions tend to be swayed by their influence rather than by whether there is an actual risk. Australia is another example of a country that is party to the Cartagena Protocol. It has approved a number of GMOs under the auspices of the Gene Technology Act. The Act addresses both product and process, but in Australia the definition of a GMO is process-triggered regardless of the outcomes of the modification. So, a GMO is defined as an organism that has been modified by gene technology, that is, any technique for modifying genes or genetic material, or anything declared under a regulation to be a GMO. This is a very broad definition that only specifically excludes mutagenesis and protoplast fusion. Because of the process-based nature of the definition, exclusions have to be based on specific techniques, rather than on the properties of the final organism. This is causing some difficulties for Australia, and they are currently considering which, if any, of the new breeding techniques should be specifically excluded. The problem here is that every time a new technique develops, a specific exclusion might be necessary, which could become complex to manage. Argentina is an interesting country, which is party to the Cartagena Protocol, and was early on involved in the development of the protocol. It's a large producer of GMOs. GMO regulation is governed by the Regulatory Framework for Agricultural Biotechnology, which was introduced in 1991. Its definition of a GMO is similar to that of the EU, but it has a stronger emphasis on product over process. Because Argentina is heavily dependent on exports of commodities, it has introduced a so-called mirror policy, whereby approvals are dependent on prior approval by its major trading partners, particularly the European Union. Argentina has been proactive in putting in place guidelines on regulation of some of the new breeding technologies, and in fact it's the only country to have done so to date. The policy states that if the product incorporates a new combination of genetic material, it would be considered a GMO. Using this definition, some of the applications of genome editing using site-directed nucleases would be excluded from regulation, as well as some applications of oligo-directed mutagenesis. However, cisgenesis would be included, although at the moment some other countries are currently recommending that it should be excluded. Moving now to Africa. South Africa is the most advanced country on the continent in dealing with GMOs. The South African legislation defines a GMO as an organism whose genes or genetic material have been modified in a way that does not occur naturally through mating or natural recombination or both. While there has not previously been clarity, it would appear that the reference to what may occur naturally implies a product-based trigger. The Academy of Science of South Africa was commissioned to produce a recent report on the regulatory implications of new breeding techniques. This comprehensive report confirms the product-based trigger and proposes a decision tree for determination of GMO status. I give a link here where you can download the full report. The decision tree is shown here and effectively classifies a product as a GMO only if heterologous genetic material is in the final product or where heritable genetic changes were introduced that could not have originated naturally. At this stage, the report and its recommendations have not yet been adopted as official policy in South Africa. Other countries in the Southern African region have followed a variety of routes. Mauritius and Zimbabwe have similar GMO definitions to South Africa, so might be able to follow the South African position on new breeding technologies. Swaziland also uses the term in the South African legislation does not occur naturally. On the other hand, countries such as Namibia, Zambia and Mozambique have process-based definitions similar to the Cartagena Protocol and therefore might have more difficulty in determining a way forward. We can, however, define some general principles that countries should take into account. Firstly, regulation should be proportional to the level of risk, so that low risk leads to low regulatory oversight, whereas high risk leads to high regulatory oversight. This means that it is the risk characteristics of the product that are the important factor to consider. Then, if the risks are the same, the controls and regulation should be the same, regardless of the process by which the product was obtained. And a quote from the recent report from the US National Academy of Sciences is very pertinent here. They said that any attempt by regulators to define the scope of a regulatory system through the definition of specified technologies will be rapidly outmoded by new approaches. This is actually the essence of the issue that regulators have to deal with, since simply deciding to exempt certain techniques or not from regulation is always going to be a stopgap measure. So, finally, we need to consider that in many countries, scientific institutions in particular are recommending a change from process-based to product-based legislation. African countries need to heed this and to consider what type of legislation best meets their needs, bearing in mind that some of the new breeding technologies offer real potential to deliver improved African crops and contribute towards food security. Thank you.